So good morning, everyone. Good morning from beautiful BC. My name is Claire Lynch, and I am the Executive Director of Clear Alzheimer's. And I'm just thrilled this morning to welcome you all to the first of our new webinar series, which we are calling Making Alzheimer's Research Clear, pun intended. So this morning, as I said, our first in the series, and the purpose of this is to bring together our donor community from all the reaches of Canada, to bring them together to hear firsthand about how their support is enabling us to support cutting-edge research, scientific research into Alzheimer's treatments, mitigating treatments and a cure. So I want to tell you briefly who we are. We are a very small nonprofit. I like to think of us as small but mighty, based in BC with a very big mandate. And that is to find a cure for Alzheimer's disease through funding scientific research into treatments and a cure. Simple but powerful. We are very proud to be able to keep our overheads as low as possible so that 100% of our donor dollars goes towards supporting the work of research. Without further ado, I'm going to introduce our panelists for today. I'm delighted to introduce Una de Boer, who is a director with the CLEAR board. She is going to be our moderator today. Una is not only a director, but has been our fearless market leader for the last several years. We owe her a great deal for how far we've come. And without further ado, I'm going to hand over to Una to introduce our guest of the week, Dr. Amanda Orr. Over to you, Una.
Thank you, Claire, and thank you everyone for signing up to hear Amanda. I promise you'll hear more of Amanda than me. I am delighted to be able to introduce Amanda. She's a great example of the type of scientist that Clear are able to fund thanks to the donations of our generous donors and supporters. So Amanda is the recipient of the Research Trainee Award, which is funded by Clear and Michael Smith Health Research BC. And that is actually what brought her to come and live in beautiful BC. So welcome to BC, Amanda. Amanda's studying at the University of Victoria, and she comes to us with a slightly different background. She's actually, her first undergraduate degree was in forensic science, and then she did her master's in forensic science and transferred into a PhD a few years back. And she has her PhD in environmental and life sciences with a focus on forensic biomaterials. And it's those biomaterials that we're really going to hear about today. She was clearly an excellent student and received the Governor General's Gold Medal from Trent University for her PhD. So congratulations, Amanda. We're delighted to have you with us because I think your philosophy speaks a lot to what we want to encourage as Clear, funding a variety of different researchers and different projects, because you're not a molecular biologist, but you have this sort of slightly different multidisciplinary background in sciences. And I know that you've said you believe that it's the collaboration that different backgrounds in science can bring to this problem to try and figure out how do we solve and create treatments and cures for dementia by, and you believe that'll be because you bring many different elements together. So I think we share that thought. Your Clear-funded postdoctoral research focuses on generating three-dimensional bioprinted models of Alzheimer's disease. That sounds very complicated, and I'm glad you're going to explain it. I've seen a little bit behind the scenes of what you're doing, and it really is incredible and mind-blowing how technology is helping and what you're doing to push that forward. Now, those bioprinted models, they mean that the drug screening process can be brought forward because you're able to identify potential treatments using these models much more quickly for Alzheimer's disease. Now, you focus, I know, on Alzheimer's disease, so it seems a good place to start to talk about. We talk about dementia as an umbrella term, and Alzheimer's disease is the most common form of it. But dementia isn't a normal part of aging, is it?
No, it's not. And so first, thank you for having me on this webinar. I'm very excited to be here and extremely thankful to Clear for co-funding my research because without them, I wouldn't be able to do this. But yeah, dementia is actually a group of symptoms that a lot of people think it's a disease, but it's usually the result of some sort of disease, such as Alzheimer's disease, being the most common form of dementia. And dementia is not a normal process of aging, and a lot of people think it is. It's just part of getting older, but it's not, because it gets progressively worse over time. And it interferes with daily life. It affects things like memory and thinking, judgment, language, and even personality. And so it is getting progressively worse as an individual, or over time, sorry.
Um, so there's clearly a very, we can see there are changes that happen in the brain with people who have dementia. Is it, would you like to explain the link to genetics? There are proteins in the brain that make this a progressive disease. Could you explain a little bit from the early onset symptoms forward, some of those changes that are happening?
Yeah, I actually have I'm a visual learner, so I'd like to share this if I can. And hopefully everyone can see my screen really
Yep,
well
I
there.
can. I think it's
Perfect.
working perfectly.
So yeah, as you mentioned, there is genetic risks associated with developing Alzheimer's disease. There are 3 genes that are linked to early onset Alzheimer's disease, so individuals typically between the age of 30 to 50, but there's also genetics linked to late onset Alzheimer's disease as well. And when we look at a healthy brain, I've just shown a few of the different cell types involved or that are in the brain. So we have our astrocytes, which are our support cells in the brain. We have microglia, which are our immune cells. And most people are probably familiar with neurons. These are the cells that really drive the communication and all of the functions in the brain. And in a normal, healthy brain, these are all functioning well and able to communicate quite nicely. However, when we look at an Alzheimer's brain, there's a significant difference. The first being the formation of these amyloid beta plaques. So these are caused by the misprocessing of certain proteins in the brain that cause these sticky plaques to form that interfere with the signaling between the neurons. So the neurons can't communicate and they can't receive nutrients, which then in turn leads to cell death. We also see things called neurofibrillary tangles. And there's this protein called tau protein, which stabilizes the microtubules throughout the neural cell. Most prominently, these are in the axon or the body of the neuron. And when these tau proteins become misfolded, they start to disintegrate, and these tau tangles start to form, which forms neurofibrillary tangles. And all of these processes, again, interfere with neural cells' ability to communicate and receive nutrients. But we also see other cells. So we have our immune cells start to become overactive. They increase the inflammation in the brain. We start to see our astrocytes undergo something called astrogliolysis, which starts to become really toxic to the neural cells, and they can start actually killing the neurons. And overall, what we see is a change in the brain mass. So we see a significant decrease in the overall size of the brain, increased ventricles. And this is when we look at, brain scans, we can actually see these changes quite distinctly when we look at the individuals who are suffering from Alzheimer's disease.
So, do you want to sort of start getting into some of your research itself right now?
Yeah, I'd be happy to do that if, yeah, we think that's a good spot. I like to talk about it. So
Yeah. So why don't you take us through
yeah.
and really sort of explain, um, as a researcher, what is it that you're looking into? How does that background that you brought, we've got some great videos to share.
Yeah, so before I get into the Sherry, I will say I'm so thankful to have this multidisciplinary background. I think it really sets me up nicely to be able to adapt and think about all these aspects of not just, biology, there's chemistry involved, there's physics involved, and how can we incorporate these into our models? So I'll go back to my screen again here. And so again, we love pictures. So I work my 3D bioprinted models rely heavily on cells. And this is something that we work with induced pluripotent stem cells. And a lot of people think when they hear stem cells, they think embryonic stem cells, which obviously has ethical considerations. But this isn't actually the case in the cells that we're working with or the stem cells we're working with. We can take cells from patients, so people who have Alzheimer's disease, we can take their cells, and this is skin cells or blood cells, and they can be reprogrammed into induced pluripotent stem cells. So these are stem cells that can form any cell type in the body. And we can start them on a neural fate. So because we're working with the brain, we're looking at using something called neural progenitor cells. And so the reason we like these is because we can create a huge bank of cells that we can use to print hundreds of models. And this is really quite a nice feature about these cell types. We can also use them to form different cell types found in the brain, thanks to the addition of these growth factors. And so this process looks pretty complicated, and it is, but we're mimicking what's happening in the brain through embryonic development. So we're able to form functional neural networks naturally through the addition of these growth factors. And what this looks like in terms of 3D bioprinting is we can take those neural progenitor cells and combine them with natural polymers. So these are polymers or materials that are found in our own bodies or found in nature. So typically from plants. And we can combine these to form our bio ink. And it's just like an ink that you would think of like a printer. So instead of us printing images or words on a page, we're printing specific shapes that we tell the printer. We pre-design it and say, we want this many layers and this size, and it will do exactly that. And not only that, it'll put those cells in the exact spot that we want them to be, because we have our cell laden bio ink. And so we can really create quite complex shapes with this, and they're reproducible, and we can achieve, again, hundreds of these in a day if we have the energy to do so. And so for us, it looks something like this. We're creating more of a three-dimensional environment for our cells, and we've actually seen literature where the cells behave very differently in a 3D environment versus a 2D. So it gives us that advantage to just culturing cells in a culture plate. And so this is something we would see for our 3D bioprinted brain models, and we can add in those same growth factors that I mentioned on the previous slide to then differentiate and mature those cells into a functional neural network. And so this is, again, mimicking what's happening in the brain naturally. And this opens up a huge avenue for us to be able to study what happens. And this also happens with Alzheimer's disease. So if we take patient cells, so individuals who have Alzheimer's disease, and we print those, we see those same proteins form in our models. So we start to see the neurofibrillary tangles and the amyloid beta plaques. And the key to this is that it happens on the timescale of a few weeks to months. So instead of waiting for someone to develop Alzheimer's disease late in life, we can see this right away in our models.
So are you saying that you can take cells from a healthy person and create more of them and print them, and cells from a person with Alzheimer's, and the outputs look different?
Very different, yes. Yeah.
That's incredible.
Yeah, it really is. And it also gives us nice controls. I'll get to it in just a minute on how it really applies to my research, but it gives us an option or an opportunity to look at how drugs will affect healthy cells as well. Is it specific to Alzheimer's cells, or how does it keep the non-dysfunctional cells happy? Yeah.
And so the biggest benefit of being able, I mean, and there are many benefits, and I know you're going to talk about it, but it's the speed that you're able to to have this sort of bank of material that you can then do tests on. Is that
Yeah.
the big benefit?
Absolutely. Yeah. We can print, and I can start this video here to show kind of, this is sped up a little bit. And it's not the exact shape that we do for our models, but it is kind of a nice pattern. But we can print these rapidly, and this would be something we can print them right into our culture dish with media, and we can have the cells immediately put in the incubator right after. And so we can print this in any design we want. And there's a lot of literature into different patterns that we use, how it affects cell viability. And basically, we want to be able to have our cells get access to those nutrients from the media to keep them growing and maturing over time. And we can use this to create pretty much any shape that we want to, that our materials will allow us to be able to do. So this on the screen here is an example of typically our half mini brains that we can print. We can also do more complex shapes such as ears. We have in our lab focuses on skin models and cardiac models in addition to our brain models. And so how we're using this is, as Una said, is we can take them and we can print, hundreds of them in one day, and we can put them in these plates that take up about the size of my hand, and we can rapidly screen them. So we can treat them with various different drug therapies and drug treatments and respond or measure the response to see, are they clearing the amyloid beta proteins? Are we seeing changes in the neuron functions and the cell functions, to see how they affect healthy cells and disease cells? And then we can use this to screen potential drug treatments much faster than what we're currently able to do with animal models.
And one of the benefits of that is you fail more quickly, right? And failing is still learning. So
Yeah.
you learn what doesn't work and are able to eliminate it. Is that an important part of what you're doing?
That's honestly the key to it all. When we think about drugs, from the time they're developed to the approval through to get them used in clinical trials, this takes years. And, if we're screening them in animal models that don't typically mimic what happens in our brains, we might miss things. And in fact, we do see a really high drug failure rate. Most of the drugs that go to clinical trials have over a 98% drug failure rate. And with these models, we can potentially screen them better because they're better able to mimic the brain. So we can right away see whether or not there's indicators that they're working well or not so much. So we can start to deviate our resources into other treatments that will make differences in people's lives.
It's incredible how quickly things are changing.
I can stop sharing now.
So, Amanda, we understand a little bit there about what you're actually doing with the 3D printing. Can you explain a little bit more? I mean, it's inspiring to see the progress you're making. What are other future discoveries that you're excited about?
Yeah, so to switch gears a little bit from just my research in general, I'm sure most people are aware of the new FDA approval of the blood tests to detect biomarkers of Alzheimer's disease. This is an incredible breakthrough because we don't have to do the previously spinal taps, quite invasive. So it allows us to do routine drugs or screening, through blood testing to be able to detect Alzheimer's disease. So we're moving into an area where we're starting to know more about it, which makes us able to detect it earlier. Also with the advancements in artificial intelligence, so AI technology, they're also using these to be able to better screen the brain scans that I had mentioned earlier. So it's better able to detect early signs of Alzheimer's disease in the changes in the scans themselves, as well as using those same AI technologies to predict patient outcomes in terms of treatment. So taking patient data, and it can create all these complex models to be able to predict how an individual may respond to a certain treatment. More related to my research, on the cell side of things is we're becoming more aware of not just the neural cells. Most of the research had focused on the neurons because of the amyloid beta and the tau proteins and their association with the neural cells, but we're looking more into the immune cells, so those microglia, and how they're playing a role. And this opens up a whole new avenue of treatment options that we can investigate to be able to say, or to see if there's something else we can use to try to create more drugs on the market or more treatments on the market to be able to make a difference in people's lives.
So where is it that you think your research is going to go next? So you've got you've got the printing. What happens now?
Yeah, that's a great question. Well, I will say I talked about earlier creating this big bank of cells, and that's really what I've been doing for the past year is trying to get enough cells for us to be able to print all the models that we need to. And this does take some time to do this, but we're now, I'm very grateful to have a group of Alzheimer's researchers in our lab and Dr. Stephanie Willer's lab. And we're ready to start testing these models to see how fast we can see the Alzheimer's phenotype. And so how fast can we see those amyloid plaques in the tau proteins? There is another colleague of mine, Dr. Jolene Phelps, who is doing some really interesting research with these tiny little vesicles called extracellular vesicles. So these are membrane-bound particles that the cells release. So they were releasing, lipids and DNA and proteins, and it's how cells communicate with one another. So they get excreted by one cell and taken up by another. And she's looking at how we can use those to treat Alzheimer's disease because there is incredible research in this area. So it's not drug-based, it's cell-based. And they can pass the blood-brain barrier, and they're showing the ability to increase cognitive benefits or cognitive functions. It's decreasing oxidative stress, it's decreasing amyloid beta and tau proteins, and really it's showing quite promising results for the treatment of Alzheimer's disease. And so we're starting to, once we have our models ready to go, we can use her extracellular vesicles to test in our models, which we're really quite excited about.
So that collaboration that I spoke about at the beginning, it sounds like there's a sort of a community of you in the lab there working away at this.
Yeah, we're really quite fortunate to have all of us from very different backgrounds. And this is research is really big, right? So it's hard, and not one person can do it. So we're very fortunate to be able to collaborate with each other on different components and take different characterization techniques, so I can work on some aspects and we can bring it together to really understand and use the knowledge we're getting from one project into another area of our research.
That's why it's so important that there are centers of excellence for this, so that you can start transferring that knowledge. Do other people in the lab use 3D printed brain cells that you're producing?
Yeah, we actually have our group, or the three of us are focused on Alzheimer's disease. We had one student who was working on Parkinson's disease. We have another student who actually is using artificial intelligence to be able to predict which combination of our biomaterials work best to mature the neural cells. So that's some really interesting research from another colleague of mine. We're pretty much the only people in our lab that work on the brain models. In terms of research, Dr. Stephanie Willard has her company Axolotl Biosciences, and they produce bio ink that they can that anyone can purchase to be able to do brain models. But yeah, we have a lot of other skin models and cardiac models as well.
So is there an area of research that you are particularly excited about moving forward?
I'm really excited to see how our models start to perform. But again, the extracellular vesicle based research is really quite exciting. They're showing such benefits in animal models that I'm really curious to see how they behave in ours. There's also, again, we're starting to see more and more research get pieced together about components that we didn't necessarily understand about Alzheimer's disease. And so we're starting to get the picture of what is happening in the brain. And this is really quite exciting to see all of this come together.
I think it's very exciting. And, I think it's important that we recognize how you're able to move things forward quicker, because the speed to be able to get those things out to market is going to be so essential as we see the volume of people who are suffering from Alzheimer's and other forms of dementia increase, partly because of demographics. But I think that brings me into some questions we've received from our audience, which are a little bit more general. And so we're going to lean in on your understanding, not necessarily always about your particular science behind it. So is there anything else about the science you're doing before we move into the next section, which are questions that you'd like to describe?
I can't think of anything off the top of my head. I would encourage people to, stay up to date with this research. It's not just our lab that's looking at this, or other labs that are looking at just neural models in general for other neurodegenerative diseases. So I hope to see some big things in the future. Yeah.
So moving into some of the questions, there have been questions about how what are the primary causes of dementia and risk factors. Is that something you could speak to?
Yeah, absolutely. So we're getting more of an understanding of what actually contributes to Alzheimer's disease, especially in terms of risk. There's actually quite a list. I believe there's 9 factors, I think, that are shown to be high risk. Vascular health is one of those. So, how healthy the blood is. So high blood pressure, high cholesterol, and diabetes can all contribute to developing Alzheimer's disease and vascular dementia. And this is because it has to do with these conditions will damage blood vessels in the brain, which then increases the risk overall. Lifestyle is another huge risk factor where individuals who aren't physically active, socially active is another area, or who have a poor diet, or who participate in activities such as excessive alcohol consumption. These are all risk factors for developing Alzheimer's disease, as well as brain injuries and inflammation. So traumatic brain injuries have also been shown to increase the risk.
Are there different types of research that are looking at those different risk factors?
Yeah, there are. We haven't looked at them in our models, but there definitely is research out there for that.
I know that we've previously had scientists speak about the brain injury risk, which is, given it's hockey season, right? The Stanley Cup, we see the concussion impact that some of our sports people suffer, and really makes a big difference.
Yeah.
So one of the questions we've got is, I think we're anxious to see a cure. We're investing in research to try and bring that forward. So one of the questions that's come in is about how quickly do you think we're going to start to see an impact of the science that's underway?
Yeah, I think that is probably the most difficult question to answer out of all these. But I do think we're seeing impacts each and every day in just the research where it might not seem like much when you have, oh, this gene, makes a difference, or this protein, or this cell type, but we're slowly starting to piece everything together with each bit of research. And this is where continued research is so critical, because we don't fully understand what happens with Alzheimer's disease yet. But slowly we're getting the picture more clear, and pun intended on that one. But it's, especially with our research, is the ability for us to save, years of research into drugs and treatments that aren't going to make a difference is a huge impact. And that impact goes back to, millions of Canadians who are currently being affected by this disease. And if we can find something, and again, allocate our resources to other avenues and other treatments, this impact will be significantly sooner than people think.
If you had to guess, because we see in the news statements about new treatments coming out all the time. But do you get asked, and do you dare to answer? Do you think there'll be a cure in our lifetime?
I hope so. I think we're really reaching this interesting, influx of technology and science and collaboration that I think that it might not even be something that we have even invented yet. But I do think that we will. And, I'm a hopeful person just because of my, I have parents and, they're aging and, I like to think that they'll be able to, if they do develop signs of dementia and things like that, that we will have effective treatments in place to be able to increase their livelihoods and make them be able to have their own identities and dignities and things like that. So I'm very hopeful, and I hope that this sparks, if there's any researchers on this webinar, that if you're not necessarily in Alzheimer's research or neurodegenerative, maybe start thinking about it. I was a forensic scientist and I fell in love with this research and my ability to contribute to the field. And so sometimes it just takes thinking outside of the box to be able to really break through.
And I think it takes funding. When one of the reasons I'm involved with Clear is you see the difference, all of the cancer research made into cancer outcomes and how dramatically and quickly treatments have improved over the past couple of decades. Mm-hmm. And I think with that same focus on Alzheimer's and dementia, we can rapidly see, if not cures, treatments that can be deployed. So it's great to see that. How widespread is the research in Canada into Alzheimer's?
Yeah, it goes coast to coast. So it's really incredible to see all of the research across Canada in Alzheimer's research. I know I'm a little biased because I live in BC, so we actually have a lot of great research happening, even just in BC alone. But Nova Scotia and Quebec and Ontario, there's all of these different universities and groups contributing to Alzheimer's research.
Do you talk when when you're speaking about what you do to people like your family? Did you think that you would find yourself in this area? Was it something that kind of from your early childhood? I mean, you started in forensic science. It's a very different path.
It's very different. No, and I don't think they know what I do, if I'm being honest.
I took them watching.
Yeah, they I didn't think so. I just, I was fascinated by forensic science. I watched CSI like most people, and I couldn't believe that it was a science that you could study. And then when you get to it, you realize that there is actual science in forensic science. And so, and it just, my drive for curiosity and trying to understand these complex systems really just keep me going. And I was a little naive at the beginning, thinking that I could go from designing synthetic blood to designing synthetic brains. I thought it would be an easy transition, and it is quite complex, but it's really interesting to be able to contribute to this research. And knowing that this research will impact so many people is just more of a reason to keep pushing to try to find some sort of cure.
So when we think about the amount of research and how much is still not known, I mean, it is incredible to me that the brain in general is still a huge area of discovery for us. And dementia and Alzheimer's is just one part of like brain function and brain malfunction, I suppose you could say. Where do you see the gaps in our knowledge that we need to be researching, we need to be understanding better to be able to make the progress?
Yeah, that's a great question. And I do think there are obviously gaps in what we know, and more so, as I mentioned earlier, that, how our immune cells play a role is a big area of research that people can be looking into and maybe are looking into right at this moment. Understanding how different components play into the development of these diseases is really crucial. And I think that's where these models, these 3D printed models, come into play, because we can mimic what happens in the brain from embryonic development, and we can do this pretty fast. So it gives us a really unique opportunity to be able to study what's happening on the molecular level. But I think, yeah, just trying to piece together what other cells, what communication's happening, how are all of these different genes affecting the development of this disease.
And is there an area that you're most excited about discovering more of?
I'm really excited about the immune cells personally. The microglia are fascinating cells in the brain, and we know a lot about the neural cells and how those are functioning, but we're actually even discovering new cells that could potentially be contributing to Alzheimer's disease. So I think that kind of area and looking at how neuroinflammation plays a role is really, really going to be an exciting area.
Well, we'll look forward to it. And we currently have a process where we invite scientists to come and apply for funding, and hopefully we're going to get some great new projects. We fund a number a year, and maybe some of them will be in these new areas, and we'll be able to help move the science forward. Amanda, I've really enjoyed speaking to you today. Thank you. I'm amazed by that printing machine. It's like science fiction, seeing cells on a test, on the plate there. So thank you for sharing that, giving us a little peek behind the scenes. And it's great to see you making the progress you are. Really appreciate it. Thank you.
Thank you so
pass
much.
back to Claire.
Thank you, Una. And especially thank you to Dr. Amanda Orr. That was absolutely fascinating. I'm sure I speak for many on the call when I say that I've learned a great deal more than I knew before this webinar. And that's what we hope these webinars going forward will do for us. And just to the audience who's still with us, know that Amanda is one of about 7 scientists who are currently funding, doing incredible work to move the envelope on what we know about Alzheimer's disease and testing out potential treatments and ultimately, of course, the cure. I have to say I'm left with huge hope. I'm left inspired. I'm left hopeful to hear that this might even happen in our lifetime. There were a few takeaways from me. I just want to share. I love the idea of the collaboration across disciplines, and I feel very proud that is something Claire supports. We encourage, and as far as possible, we support collaboration between research teams so that new discoveries are being shared around, and we can all take advantage of them in shifting the needle. I have to say that I give myself some credit. When I heard that Dr. Orr was a forensic scientist in a previous career, I knew this would be an interesting conversation. And it's wonderful that our scientists bring multidisciplinary backgrounds to what they do. And really, as in so many ways in life in general, it's all about asking the right questions to get to the right answers. And I do believe we've reached the stage in research into Alzheimer's where we are beginning to ask the right questions. I don't think that's always been the truth, but I think we're there now. And that's why it is probably within our lifetime that we'll crack this mystery. As you heard, again, continued research is so important. We've got to keep our foot on the pedal. We've got to keep our scientists funded, keep them at work, moving the needle on what they know about this dreadful disease, remembering that Alzheimer's isn't just about the patient who suffers, it's also the family it impacts, and it is society in general who carries the burden of this terrible disease. We have everything to gain by investing in continued research into this field. And so I would just ask at this point for anyone on the call who's not yet a donor that you might consider becoming a donor with CLEAR. Our acronym stands for Canadians Leading Alzheimer's Research Canadians Leading Alzheimer's Research and Leading Edge Alzheimer's Research. Let me get my spelling right, because that's all we do. And every dollar you donate goes directly to funding our scientists like Amanda. So please, consider becoming a donor today by going to our website, www.clairealzheimers.ca, and looking for the donate button. And as in a big wrap up, just to thank Una for her moderation today, for making this an interesting conversation, for Amanda giving us some of her precious time to give back to what we do. And finally, to our audience for joining us today, please spread the word. We want to do this again several times a year. We ask that you would give us some feedback on this event. There'll be a brief survey at the end of the call. I think it's three questions. Give us your responses, please, and feel free to reach out to us with your candid feedback and suggestions of what we might cover in the future. This is your platform. This is a place we would like to meet 3-4 times a year under the banner of making Alzheimer's research clear. And we just want to build this community in this space where we can get to better understand what is happening in Alzheimer's research and how we can help. So with that said, thanks to everyone, and particularly to our background crew. Alicia, you know who you are. Thank you for putting this all together. She has been has paid attention to all the details and made us all look very good today, and we wouldn't be here without her. So thank you to the production crew, shall I say, and to everyone on screen. And with that, wishing you all a wonderful day. Let's see you again soon. Bye-bye now.